Welcome to Click for IB Biology. Here we're looking at the transmission of a nerve impulse along the axon, which is called nerve propagation or impulse transmission. There's a blank version of this presentation, a PDF, that's down in the description. That's linked below. There's also 10 questions. You can test yourself with those and secure your understanding by looking at the answers. So let's take a look at the nerve propagation. So just to create the understanding of what's taking place, we've already discussed how the nerve impulse is initiated from resting potential to threshold by depolarization in the soma, or the cell body of the motor neuron. And the location of where we begin our discussion is at the axon hillock, where we've now achieved a depolarization value of minus 55 millivolts. That's a membrane potential. Membrane potential is a charge difference across the membrane of minus 55 millivolts, and this is at this location here. So now what we're going to discuss is how this generates a wave of sodium ions along the axon, along this yellow part here, all the way down to the synapse. So let's take a look at that mechanism. So students should continually track the changes in membrane potential using these diagrams here, this graph, which is created using an oscilloscope with a reference electrode in the external, the extracellular fluid here, and one recording electrode here in the axon. So we're at minus 55 millivolts from threshold. Review the previous video to understand how we've got to that point. And here comes the wave of sodium ions from the axon hillock. And as they go down the axon by diffusion from a high concentration to a low concentration, they will carry this membrane potential of minus 55 millivolts into proximity with these structures here. These are channel proteins sitting across the membrane of the axon, and these are called sodium gates, or to be technically correct, these are sodium voltage gated channels. These protein structures, when encountering the minus 55 millivolts, what this really means is that there is an accumulation of positive sodium ions around the protein. And this causes a shape change in this protein, and that opens a pore from the outside of the cell to the inside of the axon cell here, like so. And sodium ions in the fluid around the tissue fluid around the axon will diffuse through the pore into the axon here, the axon plasm. And this is illustrated by the purple section of our curve here. So we would go from minus 55 millivolts rising up past zero and reaching a peak of plus 30 millivolts. Positive charge now created in the axon plasm, a high concentration of sodium. Let me just add that there, a high concentration of sodium ions in this location here. And that's there. Now, at this point, an interesting feature happens is that the pore, which has till this point been open, is now deactivated. If one reviews the structure of sodium gates in the membrane part of the syllabus, you will encounter there descriptions of how the sodium voltage gate has three phases. There's a phase where it is open, as in depolarization. Deactivated is when a structure called, if we just draw this is the sodium gate again, and there's a thing called the chain and ball, which will then swing into position at plus 30 millivolts and lock off the channel. So we describe it as being deactivated. And then the third phase is when it's actually closed, which does not require the ball and chain part of the protein to do so. But it's worth reviewing that in the membrane part of our course. So where have we got to? We're at plus 30 millivolts. That means that we have reached the action potential. Wherever there's plus 30 millivolts, we call that the action potential, and that, of course, is the position of the nerve impulse. Now, what happens is that these sodium ions will now diffuse forward and down through the axon and accumulate around the next set of sodium gates, sodium voltage gates. And once we reach minus 55 millivolts again, these gates will suddenly open, allowing another wave of sodium ions to enter the axon, and the story will repeat itself. Now, this point here about the threshold is that the gates will suddenly open in this state here, and this is very much what is known as an all or nothing event, which means that at threshold, if we reach minus 55 millivolts, the gates will open. There will be a shape change and they will open. But if we're beneath minus 55 millivolts, they will not open and nothing will happen. So the nerve impulse is now rolling on down the axon with a repeat of this story. At that same location where we have gone to plus 30 millivolts, the sodium gates have become deactivated. What needs to happen before we can transmit another nerve impulse is that we have to do what's called repolarization. We have to get back to threshold levels. This is achieved by, at plus 30 millivolts, not only do we have a deactivation of the sodium gates, but we have an activation of the potassium voltage gated channels, shown here in orange, and they open. Potassium ions in the axon plasm are able to diffuse out into the surrounding tissue fluid, and of course this brings the charge back down, and in fact it goes all the way back down to approximately 80 millivolts below our target, and this is called hyperpolarization. So this is the function of the potassium voltage gated channels. At minus 80 millivolts, these will also deactivate in a similar cycle to that described for the sodium gates. Let's take a look at hyperpolarization. From hyperpolarization at minus 80 millivolts, the return to threshold takes a relatively short period of time, but a significant period of time that we'll discuss in the next slide. Going from minus 80 millivolts to minus 70 millivolts is performed by the sodium potassium pumps, and also again by our potassium leakage channels, which returns us to resting potential. And so you can see that in effect we have come full cycle in our description of the events of how we achieve the resting potential of minus 70 millivolts. Let's discuss this period of hyperpolarization briefly. The purpose of hyperpolarization is what's called unidirectional propagation. That is to say, if we have a motor nerve like this, there's the axon, there's the synapse, the direction of impulse travel is only from the cell body to the synapse. It cannot reverse. And the reason for this is that if the sodium inside the axon were to try to diffuse backwards, it would be diffusing into a hyperpolarized region. And in that case, it would be unable to reach the threshold value of minus 55 millivolts. And so that would be what we would call sub-threshold. And remembering our all or nothing principle, nothing will happen. In other words, it doesn't happen so that the impulse continues in one direction, unidirectional propagation. The other effect of this feature is to prevent overlapping action potentials. So if we draw the axon like so, there's the synapse, here's the cell body, and we have action potentials rolling down the axon like this, they can't merge with each other. And that's because if the sodium diffuses forward, because it's deeply depolarized, hyperpolarized rather, it's unable to actually reach the threshold value. It's starting from too deep a point, and so again we end up with the same explanation up here of subthreshold depolarization and with no response. So this is a way in which we prevent the or rather in which we maintain impulses going in one direction and that they don't actually overlap action potentials. Okay, so let's finish this discussion off with this summary diagram here, which I invite you to hesitate the video at this point and to just go through the sequence, which I've numbered 1, 2, 3, 4, 5, and so on. These are the important stages, and I think using your notes, you should construct notes to suit each of these stages which I've identified in the cycle of nerve propagation. Okay, so there's the link to the next video and chance to subscribe to the channel, please. That would be greatly appreciated. And here's the playlist for this long video.